Hello everyone, thank you for taking your time to watch this webinar. I'm Colin Hammond, a scientist with STEMCELL Technologies. Today I'll be discussing methods and considerations for using the STEMvision instrument to analyze colony-forming unit assays for hematopoietic cell therapy products. Transplantation of hematopoietic cells have been successfully used for over 50 years in clinical settings as a curative treatment for a wide variety of inherited and acquired blood cell disorders such as hematologic cancers, bone marrow disorders, and genetic diseases. Stem cell transplantation was pioneered using bone marrow-derived hematopoietic stem cells, or HSC, but inclusion of umbilical cord blood and mobilized peripheral blood as sources of HSCs in the early 2000s allowed for the dramatic increase in the number of treated patients, with well over 50,000 HSCTs being performed annually. But the therapeutic potential of hematopoietic stem cells is still being realized, and with the advancements in gene editing technologies, it holds a promise to extend the treatment to a variety of previously incurable diseases. Several hereditary disorders that are potentially curable by gene editing of HSCs are shown on the image here. As of 2022, over 400 patients have been treated worldwide by HSC gene therapy, and there are currently 12 hematopoietic cell and gene therapy products approved by the FDA, with many more under development. But taking a cell therapy from the bench to the clinic means that it must meet many regulatory requirements, including tests for conformity and quality, as well as demonstration of the functional potency of the product. This is particularly important to cell and gene therapy products, as there is inherent variability in the starting material since it comes from different patients or donors. This variability contributes to differences in the response during manufacturing processes and poses challenges to product reproducibility and lot-to-lot consistency. Therefore, tests for product quality need to be established at various stages of the product development lifecycle, such as post-processing and manufacturing, following transportation and storage, and prior to release. Process changes to the workflow also require assurances of maintained product quality. The FDA guidance, highlighted here, clearly states that no lot of a product should be released prior to testing, which includes tests for product purity and potency. Here, purity is indicative of product composition, that is, the presence of the cells of interest as well as any potential contaminants, and potency reflects a product's biological activity and its capacity to affect a given result, tests for which are expected to correlate with the clinical response. We will focus on the evaluation of potency using the colony-forming unit assay, which remains the gold standard in vitro potency assay for hematopoietic stem and progenitor cells. Initiated from studies starting in the mid-1960s, the CFU assay measures the number of functionally viable HSPCs in a population. It does this by assessing the ability of the input cells to proliferate and differentiate in a semi-solid medium into colonies, which are then identified based on morphology as erythroid, myeloid, or as multipotent. Examples of which are shown in the images here, and I hope you can appreciate the distinct morphology of each colony type, such as the redness of the erythroid colonies due to their iron-containing hemoglobin. An important feature of this assay is the use of a semi-solid medium, such as our methocult medium, since it restricts the movement of a cell and allows for growth to be attributable to an individual clone. This means that one colony-forming unit, or CFU, is one progenitor cell which produces one colony. Therefore, the CFU assay is a quantitative assay, allowing for the measurement of the frequency of the functional HSPCs in a given population. Importantly, decades of clinical research has found a high correlation between performance in the CFU assay and transplantation outcomes in patients, demonstrating its effectiveness as a measure of product potency. However, it is important to recognize that the CFU assay is a bioassay and, like any biological system, is prone to variability. It therefore requires implementation of standards around key parameters to reduce the sources of variability and to ensure consistent results. Regulatory recommendations to control assay variability include the use of qualified reagents with long-term availability, such as methocult medium. Standardization of practices are also recommended, by implementing standard operating procedures and ensuring adequate staff training. STEMCELL Technologies offers a variety of educational materials, training courses, and proficiency testing programs which can help support this. And last but not least is standardization of likely the most subjective aspect of the CFU assay, which is the counting and identification of the colonies. Standardization of this aspect can be done with rigorous training of manual operators, but automation is recommended as a method to reduce inter-operator bias and improve consistency while also accelerating the data analysis. The CFU assay requires counting and identifying the colonies present in each assay to give an indication of the potency of the starting material. And this has a level of subjectivity to it because colonies can overlap or have difficult to discern morphologies. And this subjectivity can introduce bias and variability in the results. Here are some example images of CFU colonies where, due to overlap, high density, and the presence of multi-foci colonies, some operators may score them differently than one another. Or here, where we have some examples of myeloid and erythroid colonies in close proximity, some of which should be scored as mixed colonies, and some that should be scored as separate CFU GMs or BFUEs. Reducing this variability for human operators in these common situations requires proper training and frequent refreshers. And a part of this can be done through enrollment in proficiency testing programs, which evaluate all steps of CFU assay performance. However, even with training, human operators are inconsistent. This becomes most apparent when CFU cultures are at high density. Here we can see some representative images of CFU plates that were initiated with increasing numbers of CD34 positive cord blood cells. And as I mentioned, if you plate more functional progenitors, you will end up with more colonies up to a point of saturation. In the graphs below from an internal study, we can see that as numbers of colonies in an assay increases, the variation in counts between trained operators increases too, indicating that operator subjectivity plays a large role as colonies start to get more crowded and have a higher frequency of partially overlapping with one another. However, use of the STEMvision instrument for automating scoring reduces this trend of variation and brings the overall variation lower than what is observed between trained manual operators, as you can see by the inclusion of the data in orange. So how does the STEMvision achieve this? Well, the STEMvision instrument uses a macro zoom lens, a CCD camera, and a robotic stage to permanently acquire high-resolution images of each CFU assay. Validated algorithms then use these images to accurately count, identify, and classify colonies from normal healthy samples of cord blood, bone marrow, and mobilized peripheral blood. And you can see an image of our STEMvision instrument here on the right, as well as an example CFU image, which has had its colonies automatically counted and classified by cell type by the STEMvision instrument's analyzer software. The identified colonies are circled with the colors distinguishing colonies of different types, such as red circles indicating erythroid colonies. The generated high-quality images also allow you to zoom in on individual colonies as shown by the example image here, which can help aid in morphological distinction. Automated analysis of CFU assays on the STEMvision instrument requires the use of methicol medium and meniscus-free smart dish culture plates. The meniscus-free smart dish plates enable more equal distribution of colonies through the well, as well as shadow-free imaging around the well edge. Internal validation of the STEMvision instrument demonstrated a high correlation between the colony counts by trained manual operators and the automated counts from STEMvision, in terms of total colonies, as well as distinguishing between the erythroid BFUE colonies and the myeloid CFU GM colonies. Additionally, external validation of the STEMvision instrument has been reported in a study published by Vellier et al., which found the STEMvision platform complies with regulatory guidelines for repeatability, interoperator variability, as well as intermediate precision and method comparison. The workflow of the STEMvision instrument uses three separate applications. The first acquires permanent high-resolution bright field and dark field images of each CFU assay well. The analyzer software uses validated algorithms to accurately count and classify colonies from the acquired images, producing a summary image featuring color-specific colony IDs, as I showed a few slides ago. As well, the software has options to produce customized reports for each assay, summarizing results, performing some calculations, and embedding assay images as desired. Lastly, the marker software enables users to inspect raw or analyzed images from any previously imaged assay, allowing for manual analysis or morphological analysis from a computer instead of from a microscope. Due to source-specific differences in colony size and morphology, separate analytical packages have been developed and validated to best perform for your cells of interest. Multiple packages can be purchased and used as appropriate for labs testing diverse sources. For those interested in accelerating your CFU assay further, automated analysis of 7-day CFU assays using our Methicult Express formulation is supported by a specific algorithm enabling the counting of total colonies. Additional software add-ons are also available to support a 21 CFR Part 11 compliant workflow with system access security policies, audit trails, and e-signature capabilities. One key feature of the STEMvision instrument is the speed and flexibility it offers to the CFU assay analytical workflow. Whereas manual scoring of CFU assays by an inverted microscope requires potentially lengthy hands-on technician time, the STEMvision instrument is capable of imaging an entire plate comprising 6 individual cultures in approximately 5 minutes. Subsequent analyses of these images can be done anytime after acquisition, requiring less than 10 minutes per plate, which can be performed by the computer in the background, freeing up technician time. The generated images can similarly be sent electronically to PIs or pathologists for review at their desk instead of requiring in-person analysis in the lab, which can also be limited by the longevity of the cultures. Adding to this last point, the permanent analytical records and associated images allows for the easy examination of results over time, which aids in ensuring conformity for clinical laboratory setting. In summary, the CFU assay is the gold standard in vitro potency assay for hematopoietic stem and progenitor cells. High standardization of this assay for workflows such as the production of cell and gene therapies can be aided and accelerated using an automated counting method. The STEMvision instrument offered by STEMCELL Technologies enables users to simply image and analyze CFU assays in a high throughput and reproducible manner. The STEMvision instrument offers the generation of permanent high-resolution images, fast and flexible analysis of the data, simplified reporting outputs, and optionally 21 CFR Part 11 compliant workflows with overall accurate and consistent results. More information on the STEMvision instrument and the CFU assay can be found on our website. Thank you very, very much for listening.